implies the viscosity. So resistance is proportional to viscosity, and high blood viscosity means there's a high pulmonary vascular resistance and high pulmonary arterial pressure. Increased RBC glycolysis increases 2,3-diphosphoglycerate. This 2,3-DPG binds to the beta chains of hemoglobin, reducing its affinity for oxygen, and that shifts the oxygen dissociation curve to the right. There's an increased P50, and right releases, so there's more oxygen released to the tissues. Hypoxia affects the pulmonary vessels differently than systemic vessels. There's hypoxic vasoconstriction. Systemic vessels dilate in response to low oxygen, while pulmonary vessels constrict. So that increases the pulmonary vascular resistance and the pulmonary arterial pressure. This can increase back pressure on the right ventricle, causing right ventricular hypertrophy. An adverse consequence of this effect of oxygen on vessels is seen in acute mountain sickness, when someone ascends too rapidly. Local dilation of cerebral vessels increases blood flow, so there's high capillary pressure. And from what we know about Starling forces, that can cause increased filtration and so acute cerebral edema. The constriction of vessels in the lungs pushes blood through the unconstricted vessels. So that increases capillary pressure there, and again by increased filtration, there's pulmonary edema. And this is called high altitude pulmonary edema. In chronic mountain sickness, the RBCs and hematocrit increase a lot, so there's more blood viscosity. That actually reduces tissue blood flow and can reduce oxygen delivery to the tissues. Hypoxic vasoconstriction is meant to be protective. Give blood to the better ventilated alveoli. In chronic mountain sickness, all of the alveoli are in a low oxygen state, so all the arterioles constrict, increasing the pulmonary vascular resistance and pulmonary arterial pressure by a lot. This can cause the right heart to fail. If this video helped you, give it a thumbs up, share, and subscribe. Thanks for watching, and I'll see you in the next one. Hi everyone, welcome to Bite Size Med, where we talk about quick, bite-sized concepts and basic medical sciences for study and rapid review. In this video, I've put together some equations from respiratory physiology. So I like to call this my equation chain link. We're going to link each equation to the next. Let's start with compliance. That's the easiest one. So lung compliance is the change in volume over the change in pressure. In respiratory physiology, there are three kinds of pressures: alveolar pressure, pleural pressure, and transpulmonary pressure. Transpulmonary pressure is the difference between alveolar and pleural pressure. There are three pressures, but there are four volumes and four capacities. The volumes are the inspiratory reserve volume, the tidal volume, the expiratory reserve volume, and the residual volume. The capacities are the sum of volumes. So the first two, that's the inspiratory reserve volume and tidal volume, is the inspiratory capacity. Add the expiratory reserve volume to them, and we get the vital capacity. Take just the ERV and add the residual volume, and you get the functional residual capacity. Put all of them together, and you get the total capacity. So that's the IRV, the tidal volume, the ERV, and the residual volume. Let's pick up the tidal volume now. The tidal volume multiplied by the respiratory rate gives us the pulmonary or the minute ventilation rate. From this, if we remove the dead space, then we get the alveolar ventilation rate. So it's the difference between the tidal volume, the dead space volume, into the rate of respiration. The physiological dead space is the tidal volume multiplied by the difference between arterial and expired carbon dioxide over arterial carbon dioxide. Alveolar carbon dioxide and so arterial carbon dioxide can be predicted with the alveolar ventilation equation. which is that alveolar carbon dioxide is the rate of carbon dioxide produced into K, which is a constant, over the alveolar ventilation rate. Oxygen can be determined from the carbon dioxide using the alveolar gas equation. It's that alveolar oxygen is equal to the fraction of oxygen in inspired air into the difference between atmospheric pressure and water vapor pressure minus the alveolar carbon dioxide over the respiratory quotient. This respiratory quotient is the ratio of carbon dioxide produced to oxygen consumed, and it's normally 0.8. The difference between alveolar and arterial oxygen is called the alveolar arterial gradient. That's the AA gradient. In blood, oxygen can be dissolved or bound. The bound portion is to hemoglobin, and the oxygen binding capacity of blood is how much oxygen can bind to hemoglobin, which is normally 1.34 ml for 1 gram of hemoglobin, into the hemoglobin concentration, into the saturation. The oxygen content of blood is the total oxygen in blood, so it's the binding capacity along with the dissolved oxygen. That's 1.34 into the hemoglobin concentration, into the saturation, plus the partial pressure of oxygen into 0.003. That's the solubility of oxygen. The oxygen delivery to the tissues is how much oxygen gets to the tissues. So it's the whole oxygen content of blood into the flow, and that's the cardiac output. Oxygen, carbon dioxide, and all these gases, they move by diffusion. The diffusion of a gas depends upon the change in pressure, the cross-sectional area, the solubility, the distance that the gas has to travel, and the square root of the molecular weight. The change in pressure is the pressure gradient, and it's a partial pressure gradient. This is Fick's law of diffusion. Henry's law is for the concentration of a dissolved gas. That's the partial pressure of the gas into the solubility coefficient. So all of this was about the ventilation portion of the lungs. But the lungs also have circulation, and that has its own physiology too. The resistance of vessels is 8 eta L over pi r to the power of 4. Eta is the viscosity, L is the length, and r is the radius of the vessels. So it's the same as vessels elsewhere. And the same formula is also applicable to resistance in the airways. Pulmonary vascular resistance is the difference between the pressure in the pulmonary artery and the pressure in the left atrium over the cardiac output. That's because resistance is change in pressure over flow. Lastly, the pH of blood. Don't forget the role that the lungs have in acid-base balance. That's the Henderson-Hasselbalch equation. pH equals pK plus log of the bicarb concentration over 0.03 into the partial pressure of carbon dioxide. So that's just my way of putting them all together. A couple of things that I have used to remember equations is one, you can use flashcards. You could also write down a list in the back of your notebook. And most importantly, practice a few problems so you'll get used to these equations. It's one of the really good things to quickly scan through on the day before your exam, so you can try and put them together somewhere. If you would like more videos like this, please let me know in the comment section below. I hope you found this video useful. If you did, give it a thumbs up, share, and subscribe. Thanks for watching, and I'll see you in the next one. Welcome to Bite Size Med, where we talk about quick, bite-sized concepts and basic medical sciences for study and rapid review. This video is on the compliance of the lungs and the chest wall. Now before I start, I thought I'd let you know that when I studied compliance, it really annoyed me, so I decided to make a video to sort of simplify it the best way that I can, and hopefully it helps someone out there. So let's get started. Now the breathing cycle is pretty straightforward. Air goes in during inspiration and goes out during expiration. During this cycle, there are volume and pressure changes. The change in volume over the change in pressure, that's compliance. So let's quickly go over what these changes are. The volume of air breathed in or out during quiet respiration, that is the tidal volume. That's around 500 ml. Now for this 500 ml to go in or out, the pressures have to change. And air moves along a pressure gradient from high to low. Between the atmosphere and the alveoli is a pressure gradient. Another pressure is in the pleural space. That's the pleural pressure. The pleural pressure is negative at rest at -5 centimeters of water. Alveolar is 0. And all of this is in relation to the atmosphere, which is 0. Now for the tidal volume to enter, the alveolar pressure has to become negative. The diaphragm contracts, the lungs expand, alveolar pressure goes down to -1, and the pleural pressure becomes more negative at -7.5. Air flows from high pressure to low pressure. So now from the atmosphere into the lungs, the tidal volume has entered. At the end of inspiration, the alveolar pressure comes back to 0. For expiration, which is passive, the lungs recoil, and the alveolar pressure becomes positive at +1. So the gradient has reversed, and air leaves the lungs. The pleural pressure goes back up to -5, and the alveolar pressure comes back to 0, and we're at rest again. So at rest, the alveolar pressure is 0, and the pleural pressure is -5. During inspiration, alveolar pressure goes down to -1 and comes back to 0. Pleural pressure goes down to -7.5. During expiration, the alveolar pressure increases to +1 and comes back to 0, and the pleural pressure returns to -5. Now the difference between these two pressures, that's the pressure across the organ, which in this case is the lungs. So it's the transpulmonary pressure. That's alveolar minus pleural pressure. It goes from +5 to +7.5 and back to +5. The change in volume over the change in pressure. So if there's a large change in volume for a small change in pressure, it means it's highly compliant. Versus if a smaller volume needs a higher pressure, that means it's low compliance. So high compliance means the lungs can stretch easier for smaller pressure changes. But the lungs can't just keep stretching. Remember, expiration was passive. It's from recoil. That's elastic recoil. So the elasticity of the lung, that brings it back, just like a rubber band. Once it's stretched, it wants to recoil back. So this is from elastic forces. And one of those forces is in the lung tissue itself. This is an elastic force. So it's from elastin fibers, which are in the connective tissue. When the lung stretches, these fibers stretch too. So they help bring it back to shape again. Another force is in the alveoli. The alveoli have fluid lining them, and they also have air. Between air and fluid, there's an interface. This air-fluid interface results in surface tension. The water molecules at the surface of the fluid try to contract. They try to make the smallest structure, the sphere, and that makes the alveoli more collapsible. So the surface tension is the second elastic force. Now what reduces the surface tension? Surfactant. If surface tension increases elasticity and reduces compliance, surfactant does the opposite. It increases compliance. So if we were to plot pressure against volume, you get that compliance diagram. And the slope of this curve would be the change in volume over the change in pressure. So that's the compliance, that's the formula. So more slope, more compliance. That means a steeper curve, which means it's more compliant. So this curve is for inspiration and this is for expiration, and they follow different paths. That phenomenon is called hysteresis. And one of the reasons hysteresis happens is because of surface tension that needs to be overcome during inspiration. So that's why the curves are different. Now all of that was for the lung, and the lungs are inside the chest wall, and it has to stretch as well. So that's the chest wall compliance. And the compliance of these two together is lower than any of the individual ones. So this is the lung and this is the chest wall. And at rest, remember, the volume in the lungs is at functional residual capacity. The lungs have a natural tendency to want to collapse, and that's an inward pull. The chest wall wants to expand, and that's an outward pull. These two forces balance each other, and the system is at neutral. When the lungs expand, the volume is getting higher, and a bigger lung wants to recoil more. So its inward pull is stronger than the outward pull of the chest wall. So as a unit, they want to collapse. and that forces the air out. Now reverse it. When the lung is contracted, there's a lower volume, and the smaller lung has lesser recoil, and the chest wall now has a stronger outward pull compared to the lung's inward pull. So as a unit, it wants to expand, and air enters the lungs. So when the unit contracts, it wants to expand, and when it expands, it wants to contract. And that is the compliance of the lungs and the chest wall. If this video helped you, give it a thumbs up, share, and subscribe. Thanks for watching, and I'll see you in the next one. Hi everyone, welcome to Bite-Sized Med, where we talk about quick, bite-sized concepts in basic medical sciences for study and rapid learning. This is a short video on surfactant and surface tension and respiration. The alveoli are the functional units inside the lung. Inside these alveoli is a fluid lining. Also inside the alveoli is air. So that creates an air-fluid interface. And whenever air and fluid come into contact like this, the water molecules that are on the surface, they attract each other and they try to form a small structure, a sphere. This is the same thing that happens with a raindrop. That air-water interface there creates that shape. So this creates tension, and that is called surface tension because it's tension at the surface. Now imagine this happening inside the alveoli. The water molecules are contracting. Air is leaving during inspiration, so it's getting smaller. And the surface tension can make these alveoli collapse. More the surface tension, more the chances of them collapsing. The surface tension is an elastic force, so it's one of those things that causes the lungs to recoil. The other thing is elastin in the lung tissue, which stretches when the lung stretches. So these oppose stretching of the lung, and that stretching is compliance. So high surface tension, low compliance. Laplace's law for a sphere is that the pressure is twice the surface tension over the radius. This is the pressure that keeps the alveolus open and is proportional to the surface tension and inversely proportional to the radius. So that means if there's a high radius or a big alveolus, then less pressure is needed. It doesn't collapse easily, while a smaller alveolus or one with a lower radius needs more pressure to keep it open, so it collapses easier. An alveoli have to be small, because that increases their surface area. Their main job is gas exchange, and one of the factors that affects diffusion is cross-sectional area. So what can we change in this equation for them to stay open? The surface tension. By lowering the surface tension, we can lower the pressure, and the substance that does that is surfactant. Surfactant is produced by type 2 alveolar epithelial cells, and it's a surface active agent. Its production starts later in gestation, around 24 weeks, and its main components are phospholipids, particularly dipalmitoyl phosphatidylcholine, which is also called lecithin, or DPPC. There are other things too, like phosphatidylglycerol, surfactant, apoproteins, and things like that. But that lecithin, that increases with gestation. So what's measured is a lecithin sphingomyelin ratio, and that ratio becomes 2:1 when surfactant matures in late gestation, by over 35 weeks. Now it's a phospholipid, which means it has a phosphate component and a lipid component. And it's amphipathic, so hydrophobic and hydrophilic portions. Hydrophobic means it doesn't like water, and hydrophilic means it does. The tails are the hydrophobic portion. They don't like the water, so instead they face the lumen when they line up along the interface. Now this interface is responsible for the surface tension. So by lining up like this, surfactant reduces the surface tension. Now remember I said it has to be synthesized, so what happens if it's deficient? Alveoli have a more tendency to collapse. That's respiratory distress. It's called neonatal respiratory distress syndrome, or hyaline membrane disease.
Wow.
So more the surface tension.
What did I do? Let's see what I learned from this guy.
Alright, engineers, in this video we're going to talk about respiration and high altitude. This is such a big deal because when we have high altitudes, for example, these guys are going high altitude, known as it can be about is how our respiration changes during the actual ascent during the high altitudes. Okay? So let's go ahead and start there. So first thing that we need to understand about being at high altitudes is what the actual atmospheric pressure. You know, when you talk about the atmospheric pressure, generally, atmospheric pressure, or barometric pressure, is approximately 760 millimeters of mercury. And out of that 760 millimeters of mercury, it's a mixture of many different gases like nitrogen, and water vapor, and oxygen, and CO2. Well, what we're talking about here is oxygen, because at high altitudes, the availability of oxygen decreases. Why? So as you go higher, the percentage of oxygen out of this pressure decreases. So normally the partial pressure of oxygen is 21% of this. So if you take 21% of 760 millimeters of mercury, it'll give you approximately 160 millimeters of mercury, about, right? Now, what's the problem? How is this such a big deal, right? Well, when you ascend to high altitudes, the availability of oxygen decreases. So the percentage of oxygen within the atmosphere decreases. So then the partial pressure of oxygen within the atmosphere decreases. So now let's look. If it's less than 21%, this new partial pressure of oxygen is going to be less than 160 millimeters of mercury. Okay? Now you might say, okay, why is that such a big deal? We're going to see here in just a second. Let's say here I have the lung, right? And in the lung, it's made up of many different alveoli. Okay, we're going to treat this whole space in here as an alveoli. Okay, we're going to treat this whole space in here. So now, let's say that the partial pressure of oxygen normally within the lung, normally at normal sea level, is approximately about 104, we're going to keep it at the average about 100 millimeters of mercury. So normally, the partial pressure of oxygen within the alveoli is approximately 100, we'll write it down, 104 millimeters of mercury. That's approximately, right? But that's whenever it's at 760 and the percent of oxygen is 21%. If the percent of oxygen decreases and it goes below 21%, now there's going to be disastrous effects. Now, some of you might be wondering, okay, I don't get it. I thought, Zach, remember in the videos you said that air moves from areas of high pressure to low pressure until equilibrium is reached. Well, if that's 160 out in the atmosphere, and this is 104 here in the alveoli, that doesn't make any sense. Shouldn't it be 106? Yeah, it should, but guess what happens? As oxygen is coming down into the actual lungs, it mixes with some water vapor. And as it mixes with some of the water vapor, some of the oxygen actually gets lost in that process. So the amount of oxygen that's actually coming down is only 104. So you actually, whenever it combines with some of the water vapor as it's moving down, you actually lose some of that oxygen. And then it becomes 104 millimeters of mercury. So if it actually drops, if the partial pressure of oxygen within the atmosphere when you go to high altitudes drops below than 160, what's going to happen to the partial pressure in the alveoli? It's going to drop below 104 significantly. So for example, let's say it dropped down to like 130, okay? So the partial pressure of oxygen within the atmosphere dropped down to 130. Then the partial pressure of oxygen within the alveoli is going to drop down significantly. Let's suppose, though, for this actual video, we're going to suppose that the partial pressure of oxygen is in the atmosphere is so low that it causes the partial pressure in the alveoli to drop down to approximately 60 millimeters of mercury. Okay? So we're going to assume that the partial pressure of oxygen within the altitude and the atmospheres, whenever you're going high altitudes, is so low, so less than 160, that whenever it moves into the alveoli, it actually becomes about 60 millimeters of mercury. Let's suppose that. Why is that a problem? Okay. So, remember the structures that we had within inside of the actual, specifically the medulla and the pons? You guys remember that? Real quickly, what do we have? This was the VRG, right? Ventral respiratory group consisting of complex of neurons, clusters of neurons. This was the DRG. This was your central chemoreceptors. This was your apneustic center. And this was the pneumotaxic center. And both of these were making up your pontine respiratory group, right? So both of these, pneumotaxic center and the apneustic center, collectively are making up your pontine respiratory group. So pontine respiratory group. Alright, cool. Well now, if you guys remember, The VRD and the DRG had very specific inspiratory neurons, right? Those inspiratory neurons were doing what? Well, they could send down, let's actually do this in orange. They would send down these actual fibers, right? They were sending down these actual descending axons. And these descending axons were coming out and stimulating specific actual, what? Cell bodies here, right? The somatic motor neurons within the ventrograde horn. And what were they doing? They were coming out and collectively forming the actual neurons that were going to supply the external intercostals, and that were also going to supply the, what? Diaphragm. And again, if it was the diaphragm, it was actually going to be via the phrenic nerve. And if it was going to be through the external intercostals, it would be the intercostal nerves. And if it was the intercostal nerves, that was at T1 to T11, phrenic nerve is C3 to C5, right? Okay, well this is the normal way. If you guys remember from the video when we talked about the control and the regulation of respiration, we talked about what happens whenever the partial pressure of oxygen is really low. Alright, so now let's take this guy over here and let's see what's going to happen, because this should make sense now. Okay. Partial pressure of oxygen within the alveoli. We're going to treat this as the alveoli. The partial pressure of oxygen within the alveoli now, when we're at high altitudes, is so low that it drops to 60 millimeters of mercury. Alright, sweet deal. But then, let's say, as the blood is coming through, what is this? Here's your right side of the heart, right? This is your right atrium. This is your right ventricle. This is the pulmonary trunk, pulmonary arteries, so we'll put PA, pulmonary arteries. And then coming out over here, what are these going to be? These over here are going to be your pulmonary veins. Pulmonary veins, okay? As the blood is flowing through the pulmonary artery, what is the pressure? What is the partial pressure of oxygen? So the partial pressure of oxygen within the blood is approximately 40 millimeters of mercury, right? And if you guys remember from before, air will flow from areas of high pressure to areas of low pressure until there's equilibrium reached, right? So what's going to happen then? Oxygen is going to flow from the alveoli into the blood. What is that called? External respiration, right? So where's the actual blood going to flow? Sorry, the oxygen going to flow. The oxygen is going to flow here into the actual pulmonary capillaries and exchange this process with the oxygen. So what should oxygen come out as? It should leave and go into the pulmonary veins as having a partial pressure of oxygen equal to 60 millimeters of mercury. Okay. What about CO2? Well, right now we'll talk about CO2 for a second here. The partial pressure of CO2 normally within this blood is going to be approximately about 45 to 46 millimeters of mercury. Whereas in the alveoli, the partial pressure of CO2 is only what? Let's do this in a different color. Let's do this in the brown also. So the partial pressure of CO2 within the alveoli is about 40 millimeters of mercury.
Yeah.
So it's approximately 40 millimeters of mercury. We'll see how this changes through this process. So again, where would the air want to flow? The air would want to flow from areas of high pressure to low pressure. So where is this going to go? It's going to go out. It's going to come out here into the alveoli. And it's going to move from the blood into the alveoli until the partial pressure of CO2 in the blood is going to equilibrate with the partial pressure of CO2 within the alveoli. So what should it leave as? The partial pressure of CO2 as it leaves should be equal to 40 millimeters of mercury. So that's what it should be. So the partial pressure of CO2 should be equal to 40 millimeters of mercury. Okay, why am I telling you all this? Because if you get this, it's going to make so much sense here. Watch this. The problem is they're having hypoxemia, right? What do we define as hypoxemia? It was basically whenever you have this low arterial partial pressure of oxygen. What did we say whenever the partial pressure of oxygen is equal to 60 or less than 60 millimeters of mercury? What chemoreceptors are getting activated? So look at this. The partial pressure of oxygen is so low. So what is it going to do? It's going to come up here. It's going to go back to where? It's going to go from the pulmonary veins. It's going to go into the right atrium. Then it's going to go into the, I'm sorry, not right atrium. It should be left atrium. Shame on me. So it should be left atrium. And it should be left ventricle. Goes up into the aorta. As it goes up into the aorta, it runs up through. Again, it can come up actually eventually to the common carotid artery. So this is the common carotid artery. Common carotid artery. This is the external carotid artery. This is the internal carotid artery. Again, internal carotid artery, external carotid artery, subclavian artery, subclavian artery. You get the point, right? And this would be the right side, that would be the left side. As you go up, around this point where the common carotid artery bifurcates and goes to the external carotid artery and internal carotid artery, there's these little bodies. What are these little dudes up here? These little smiling dudes called? Remember those? Those are your peripheral chemoreceptors. And these peripheral chemoreceptors are special. So again, what are these structures here called? They're called peripheral chemoreceptors. And again, if they're here located within the actual carotid bifurcation point, they're called the carotid bodies. So these are called your carotid bodies. And if they're located here at the aortic arch, they're going to be called the aortic bodies. And if you guys remember, these are responding to a low partial pressure of oxygen whenever it's below 60 millimeters of mercury. So let's assume that it actually is going below 60 millimeters of mercury. At least 60 or below. What is it going to do? It's going to stimulate these guys. You guys already know the mechanism. It inhibits those potassium chains, and then calcium comes in, dopamine is released. What happens to these guys then? If this low partial pressure of oxygen stimulates these guys, they're going to activate their actual sensory afferent fibers of those two cranial nerves, or the cranial nerves that were being activated in this process. If you guys remember, it was actually if it was connected to this one, if it was connected to the aortic bodies, it's going to be cranial nerve, 10, which is the vagus nerve. If it was connected with the actual carotid bodies, it was going to be cranial nerve 9, which is the glossopharyngeal nerve. If they're stimulated, what are they going to do? They're going to send signals. What are they going to send signals with? So down, if we follow, let's say that we follow the cranial nerve 10 and cranial nerve 9. As they send signals over, let's say that we follow the signal like this now. Here's the signal. And again, who's carrying the signal? It's going to be cranial nerve 10, and this is going to be cranial nerve 9. And again, what was cranial nerve 10 and 9? This was glossopharyngeal. This is the vagus. They're sending centers to the respiratory centers within the medulla and within the pons. What was the overall result, if you guys remember? Well, the stimulation was what? Let's remember what the stimulation was. It was low partial pressure of oxygen. And it has to be at least 60 millimeters of mercury or less. It is in this case. Let's assume that it is in this case. What is it going to do? It's going to stimulate specific centers within the medulla, right? What are those two centers? One is it's going to activate the DRG. The other one is going to activate the VRG. But specifically, what action of these two guys? So if we follow these guys in here, what are they going to do? Stimulate the VRG, and it's also going to stimulate the DRG. And it's going to activate having more frequency of action potentials. So what's going to happen to the frequency of action potentials? The frequency of action potentials is going to go up.
God, what the
If the
fuck?
frequency of the action potentials go up, what's going to happen to these the diaphragm muscle? It's going to be stimulated heavily and frequently. And then the external intercostal is going to be stimulated frequently and heavily. What's going to happen to these individuals? They're going to hyperventilate. Because if you're actually having increased frequency of contraction, increased frequency of action potentials, increased frequency of contractions, what's going to happen to this person? They're going to have increased alveolar ventilation. What does that mean? That means that you're going to increase the respiration rate and increase the actual depth. So you're going to increase the respiration rate and the depth. In other words, the tidal volume in this case for the depth, right? We can put that the tidal volume. If you do that, what's going to happen? If you increase your alveolar ventilation as you're going to high altitudes, your alveolar ventilation rate is going to increase originally as you're going up to these high altitudes. What's that going to try to do then? It's going to try to increase the ventilation process. So it's going to try to bring more air in from the atmosphere to the best of its ability. So as you try to bring more air in to the best of your ability from the atmosphere, it's going to attempt to increase the ventilation process. What does that mean then? It's going to try to increase the actual exchange of oxygen with the blood. But it might not change significantly. Let's say that it actually goes up a little bit, and it goes up to about, let's say, 75 or 80, whatever. It goes up to about 75 or 80. Where's my brown marker? So let's say it goes up to about 80 millimeters of mercury, okay? If that's the case, then what's going to happen to the partial pressure of oxygen within the blood after the exchange occurs? It should go up to about 80 millimeters of mercury. Okay, that's good. What happens to the stimulus now for the actual aortic bodies? It goes away. What happens to the stimulus for the actual carotid bodies? It goes away, right? and then we basically inhibit the action potentials that are going to the diaphragm, the external intercostals, as we increase the ventilation process to appropriate values. So as we try to, in other words, until we bring the partial pressure of oxygen below 60 millimeters of mercury to the best of our abilities above that, then the hyperventilation or the increase in ventilation and depth rate is also going to slow down. But there's a problem with this. Something happens that's not good. Remember I told you CO2 was moving also? And we know that it normally moves from 45 to 40. Well, guess what happens? As you move to higher altitudes, you know what happens within the atmosphere? The concentration, the partial pressure of CO2 within the atmosphere, it also decreases slightly. So let's say now that the partial pressure of CO2 within the atmosphere decreases slightly. So let's say that now the partial pressure of CO2 decreases slightly. So let's say that it drops down to about, because it's less than, let's say it's actually 25 millimeters of mercury, whatever, pick a number, 25 millimeters of mercury. If that's the case, then what's the partial pressure of CO2 within the alveoli going to be? It's going to be a lot less. So let's assume that it's so low that it actually drops to about 25 millimeters of mercury, let's suppose. Okay? If that's the case, then what's going to happen to the movement of CO2? Well, before it was 45 to 40. Now it's 45 to 25. That's a 20 gradient difference right there, right? As compared to just a 5 gradient difference. That means more CO2 than normal is going to be being exhaled out. As you try to exhale out more CO2, what happens to the CO2 partial pressure within the blood? It drops. It drops until these two pressures equilibrate. What's the partial pressure of CO2 going to be now? The partial pressure of CO2 is going to be 25 millimeters of mercury. Oh my goodness, this is really, really bad. Why is this really, really bad? Let me explain to you why it's really bad. Because if the partial pressure of CO2 is really, really low, what can that do? Let's come over here and see what happens in the brain. This is not a good thing. Not a good thing. Okay, so now we said that the partial pressure of CO2 is extremely low now because the atmosphere is low in CO2. So now the partial pressure of CO2 within the alveoli is 25. And in other words, whenever there's an exchange across the pulmonary capillaries, the partial pressure of CO2 within the arterial blood becomes 25 millimeters of mercury. That means that CO2 that's actually coming into the cerebrospinal fluid, when it crosses, what was this called here? The blood-brain barrier. When it crosses the blood-brain barrier and it combines with water, if these two combine in the presence of an enzyme called carbonic anhydrase, it'll form carbonic acid. Carbonic acid dissociates into what? Bicarbonate, and it also dissociates into protons. This area I should do this with you. And then what happens with these protons? These protons are normally trying to act on the central chemoreceptor. Well, what did we say was the problem? The CO2 is low. The carbonic acid is low. The H plus is low. What did we say happens if it's really, really low? It's not stimulating the actual central chemoreceptors. In other words, it starts inhibiting it. If this inhibits the central chemoreceptors, what does it do to the DRG? It inhibits the DRG. If the DRG is inhibited, what does it do to the actual DRG? It inhibits it. What happens then to these action potentials that are going down to the phrenic nerve and to the actual external intercostals? It's actually going to be decreasing in frequency. So what happens to the frequency then? The frequency decreases. As the frequency decreases, then what happens? The alveolar ventilation decreases. As the alveolar ventilation decreases, what happens to the rate and the depth? It also decreases. And then what happens over here, right? You're going to have very, very slow breathing. And that's a problem now, because originally, what was happening? You had hypoxemia, right? Now if you have slow breathing, are you going to be able to allow for enough oxygen to keep coming in from the atmosphere? No. That is a bad thing, and we'll talk about why, because it can lead to acute lung symptoms. So again, whenever this happens, your drive originally was hypoxemia, and then you're trying to hyperventilate to compensate for the hypoxemia. But as you try to compensate, what are you doing? You're breathing off a lot of CO2. As you start breathing off a lot of CO2, what happens? You go into a situation where so much CO2 is lost that it can produce a condition called respiratory alkalosis. Respiratory alkalosis, and basically that's when you have extreme, your pH is really high, so your pH is higher than normal, but this pH is high due to low CO2 levels, okay? That's the actual stimulus, and it's because you're breathing off a lot of CO2 because the actual CO2 within the atmosphere is a little bit lower the normal. So we changed the number. We said it was about 25 now. Okay? I could have picked on any other number. I just picked 25 just for that. Okay? There's no rhyme or reason. Okay, so again, respiratory alkalosis can be produced in this situation, which is extremely dangerous, because now, what did you do? You took away the stimulus for this hyperventilation. So now, there's two things that are trying to be able to work here. What are the two things that are trying to work? How is this produced? So whenever there was this low partial pressure of oxygen, this was trying to increase ventilation, right? But then as you increase the ventilation, you bring the partial pressure of oxygen back up. Let's assume that you bring it back up to normal. Okay? But in that process, as you increase your ventilation, what did you do to the partial pressure of CO2? You decrease the partial pressure of CO2 very, very low. What does that do to the actual action on the central chemoreceptors? It inhibits the central chemoreceptors. If you inhibit the central chemoreceptors, then what's going to happen to the ventilation? You're going to decrease the ventilation. So if you see what I'm saying here, it's basically you're having an opposed action. So there is ventilation occurring, it's just not as high as you would want it to be. It's very moderate instead of excessive to be able to get as much oxygen to the tissues as possible. And that's why it's dangerous. How does our body deal with this though? Okay, well let's say that you're a good hiker, you've done this before. As you're ascending, you decide, all right, I'm going to take a couple days, I'm going to stay here, I'm going to wait, and I'm going to let my body acclimatize, okay? I'm going to try to acclimate to these actual conditions. How does the body acclimate the conditions? We got to go to the kidney and see. So now let's see what our good old kidney does for us, all right? In our kidney, you have, this is actually, if you guys have watched it, you know that this is the proximal convoluted tubule, this is the actual loop of Henle, the ascending and the descending. This is the distal convoluted tubule, and then this right here that we're looking at is the collecting duct. Collecting duct. Okay. Now, there is these two types of cells present within our kidneys. They're called intercalated cells, and they basically are playing a role with our acid-base balance. But there's one particular thing. There's A and B cells. We're going to look at the intercalated B cells. And don't get these confused with the B cells within your immune system. This is the actual cells in the kidney, okay? Now, what happens with these intercalated B cells? Well, if you guys remember, we can actually bring CO2 in, right? So let's say here's our CO2, and the CO2 comes in here. You know that the CO2 is going to combine with the water. When these two combine in the presence of an enzyme called carbonic anhydrase, what happens? It gets converted into carbonic acid, which breaks down and disassociates into protons and bicarbonate. You know what our body does? It says, okay, let's assume that right here, I put a blood vessel. Well, actually, let's just put it right here. Even though it's a vein, it's for the purpose of understanding this, okay? Here's two options. He says, okay, the pH over here in the blood is really high. Okay. What can I do? Well, let me actually not put bicarbonate, because normally what you can do is you can bring bicarbonate into the bloodstream, right? But we don't want to do that. We want to get rid of the bicarbonate. So look what it does. It's very tricky, and it pumps the actual protons out here. And you have to use ATP for this process, right? So in order for you to do this, you have to utilize ATP and other different types of ions. So you're pumping the protons out into the blood. But then what are you going to do with the bicarbonate? You're going to excrete out the bicarbonate in the urine. So as you start excreting out bicarbonate within the urine, you're losing a lot of your bicarbonate and bringing protons into the blood. What happens to the pH, guys? The pH starts going back down to at least normal values. Oh, that's so freaking cool. All right, so that's how our body's helping to deal with this. So if you stay at the high altitude for a couple days, maybe even a week if you really want to be completely acclimatized, right? Basically what's happening, your kidneys, your intercalated B cells are coming to the rescue, and they're putting protons back into the blood, which is bringing the pH down. We'll see how that helps in just a second. And then it's getting rid of the bicarbonate. Okay, how is this fixing it? Because you might be like, what that mean, man? I got you, don't worry. All right, so look, we said that there was going to be a lot more protons. If there's a little bit more protons here, then there's less bicarbonate. So look, we got rid of a lot of this bicarbonate. If you decrease a lot of this bicarbonate right here, it's not going to be able to bind with as many of these protons. Because you know that this is basically a buffer here. Carbonic acid can break down into two things. It can break down into H plus and bicarb. This is basically your buffer. But according to a guy named Le Chatelier, he came up with this concept that he said if you decrease this side, right, then what's going to happen? You're not going to have as much of this guy over here. So what are you going to want to do? You're going to want to break down, you're going to want to shift the reaction to the right. So you're going to want to have more carbonic acid break down into protons and bicarbonate. So as you do that, what are you accumulating more of? You're accumulating more protons. Also, you're going to have a little bit more protons accumulating within the blood too. But the protons can't cross the blood-brain barrier. So the main thing to remember is that you're excreting bicarbonate. And as you get rid of bicarbonate, bicarbonate ions decreases. Le Chatelier's principle, let's see if I can spell this. Le Chatelier's principle is basically saying that as you decrease this side of the reaction, decrease the bicarb, the reaction is going to want to shift to the right. As it shifts to the right, you make more protons. More protons are going to do what to the central chemoreceptors? Stimulate them. If you stimulate them, you're going to stimulate the DRG. If you stimulate the DRG, you stimulate the VRG. If you stimulate the VRG, you have more action potentials coming down. If you have more action potentials coming down, your frequency of the actual ventilation is going to, or action potentials is going to increase. You're going to have more frequent contractions. If you have more frequent contractions, oh my goodness, you guys should get this already, right? There's going to be an increase in ventilation rate. There's going to be an increase in the depth and the actual, in this case, the tidal volume. And then what's going to happen? You're going to have this action here where you can go back into this hyperventilatory state and do what? Try to bring more oxygen back into the bloodstream and exhale some more CO2. Okay, but you do it to a level that's accommodating, it's acceptable, right? You're not in the situation to where you're in respiratory alkalosis or you're not having excessive hypoxemia. So that's an amazing thing, but unfortunately, it's not enough. Unfortunately, it's not enough. So how else could you deal with this? Well, the body has another way of dealing with it. It's really cool. All right, so let's say here, your kidneys come to the rescue again. If you guys remember, we said, if you guys watched the actual videos within hematology, we talked about the proximal convoluted tubule. And if you guys remember, the proximal convoluted tubule was responsible for secreting a special hormone. Remember there was the hypoxia inducible factor who was bound by that little enzyme there? And we said that whenever there was low oxygen, so in this case we should be specific, we should say low partial pressure of oxygen, right? Because the partial pressure of oxygen is still going to be low. It's never going to be at 100 whenever you're going to high altitudes. So your body has to compensate. Whenever there's low oxygen, you can't hydroxylate this enzyme here. So this enzyme is inhibited. And what happens to the hypoxia inducible factor? It comes and it activates specific genes. And when it activates specific genes, it produces a hormone. What's that hormone, if you guys remember? That hormone was called EPO, erythropoietin. All right? What is erythropoietin going to do? Erythropoietin is so cool because it comes over here to the bone marrow. What's within our red bone marrow? Do you guys remember that we have the red bone marrow within the epiphyses of the long bones within that spongy bone, right? It's going to stimulate the bone marrow to start producing more red blood cells, right? Because if you remember, let's say that we draw in here, what do we have? We had a pluripotent stem cell that can divide into a myeloid and a lymphoid stem cell. And then the myeloid stem cell can actually go to develop, eventually, red blood cells. Or it can go to make platelets and white blood cells, certain white blood cells. EPO, if you guys remember, it was stimulating the actual formation of more red blood cells. If you have more red blood cells, what does that mean then? If I have more red blood cells, I have more hemoglobin. If I have more hemoglobin, I can carry more oxygen. So what's going to happen to the oxygen carrying capacity? It's going to increase. What is that called though whenever you make a lot of red blood cells? Polycythemia. So in these individuals, what are you going to see with them? Not only are you going to see that they have elevated erythropoietin levels, they're going to have very high erythropoietin levels, but they're also going to make a lot of red blood cells. And whenever you make an excessive amount of red blood cells, that's called polycythemia. Okay, so they're also going to have polycythemia. Okay, so they have polycythemia, increased ventilation, increased erythropoietin production. Wow, they have a lot of things going on. Unfortunately, that's still not it. Okay, look what else is happening here. So again, we said increase. So what was the things that we said so far? So far we've said there's been an increase in ventilation. The second thing we said that there was an actual increase in number of red blood cells, which is called polycythemia. Polycythemia. Now for the third thing. Okay. Well, if you guys remember, we're trying to increase the ventilation, right? That's what we're attempting to do. So if you guys think about the alveoli like this, let's say here I draw a small alveoli, right? Here's the alveoli, and then here's a small little capillary. Let's have a small little capillary come around it, right? Okay. Right now, what have we done? We've increased the number of red blood cells. Right, we've increased the number of red blood cells. So if there's more red blood cells, what does that mean for the actual amount of blood that's coming through here then? There's actually going to be more blood flow, so more perfusion coming through, right? There's a lot more red blood cells that you can oxygenate. What else are we trying to do? We're trying to increase the ventilation. Well, as you try to increase the ventilation and you increase the perfusion, isn't that one of the most efficient ways to have good ventilation perfusion coupling? Right? Because what have we said? That ventilation over perfusion, V over Q, is equal to 0.8. If you increase the ventilation, you want to couple it. So how would you do that? Because if you increase this, it would increase this number. You're going to want to increase the perfusion, because if you increase the perfusion, you decrease this number. So you help it come back to normal range. So there's good ventilation perfusion coupling, which means what? There's efficient gas exchange. Efficient gas exchange means that you're going to have a very efficient movement of oxygen from the alveoli to the blood and a good movement of CO2 from the blood to the alveoli. That's amazing too. Oh my goodness, it's so cool. So again, increased ventilation, increased number of red blood cells due to polycythemia, and because of that, there's adequate, or we should say normal VQ coupling, which means that there's efficient gas exchange. Alright, sweet deal. One more. This is kind of the one that happens if you are at high altitudes for a little bit of a longer time period. So if you're at high altitudes for a little bit longer than normal, your body does something absolutely amazing. Okay. Let's say here's these tissues. And what's happening with some of these tissues? We said that normally whenever you're at high altitudes, you're developing hypoxemia, right? So there's not a lot of oxygen delivery normally to these tissues because the low partial pressure of oxygen. And again, this is due to the hypoxemia due to being at high altitudes. So you're not getting a lot of delivery to the oxygen tissues. I'm sorry, you're not getting a lot of oxygen delivery to the tissues. So how does your body deal with this? Okay. Inside of your blood vessels, you have these actual endothelial cells here. And here's your endothelial cells. These endothelial cells and other different tissue cells within the vicinity, other different tissue cells, start producing very amazing chemicals. One of the chemicals is called vascular endothelial growth factor. And what is this vascular endothelial growth factor? So there's that, and then there's platelet-derived growth factors. There's a lot of growth factors that are released. What is this basically going to do? It's pretty sweet. Look at this. Let's say that it secretes this, and you actually sprout another blood vessel. So coming off of this, I'm going to have another blood vessel. It also, let's say it stimulates over here. I'm going to have more blood vessels. Let's say I sprout off of this one. I'm going to have more blood vessels. Sprout off of this one. More blood vessels. You guys get the point. What am I doing? I'm making more blood vessels to accommodate for this actual situation.
Oh, the other video
ended. There's a decreased oxygen delivery.
Did it?
What is that called whenever you're making...
Whatever.
Cram.com. Welcome to another MedCram lecture. We're going to talk about hypoxemia again, and this time we're going to talk about diffusion. Again, hypoxemia literally meaning low oxygen in the blood. Let's talk about diffusion as a possibility for a mechanism for hypoxemia. To talk about that, though, we've got to sort of break down the alveolus, and I'm drawing that here, and then we'll switch to a blue color. This is to demonstrate the pulmonary artery, which, as you know, is deoxygenated, and we have the capillary. And then we'll switch to red to show that, in fact, the blood is now oxygenated. And then this green is going to be oxygen, because you know that oxygen tanks are always green. Right? Okay, so we've got oxygen coming down into this area. And then there's something that we should talk about called the capital A, lowercase a gradient. Okay, so you guys know how a lung works. John West is quoted as saying, the lungs are very, very easy to understand. Air goes in and out and blood goes round and round. Well, let's sort of break that down a little bit to understand it a little bit better. Oxygen's going to come in into the alveolus, and it's going to diffuse past this one cell layer thick epithelium, which is the epithelium of the lung, and then it's going to go through this one cell layer thick endothelium, which is going to then go into the pulmonary artery, pulmonary capillary, and then pulmonary vein, which is going to become oxygenated. The problem here is that sometimes you can have a blockage in this area, and that can cause a diffusion abnormality. Where you'd see this is particularly in patients with infiltrated diseases of the lung, like pulmonary fibrosis, especially if they exercise. Let me give you an example. If you've got a patient with fibrosis and they're at rest, so that means there's stuff in the middle here, in this interstitium, and it's preventing oxygen from diffusing in. These red blood cells, normally, to get through, take about 0.75 seconds to transmit through this pulmonary capillary and to pop out the other side with oxygen. Now, as they're going that slowly, they're able to equilibrate with the oxygen here in the alveolus. That's no problem. That means the oxygen coming out the other end is, 95% saturated. However, when they start to exercise, cardiac output goes up, and even though there are parallel systems that the blood can go through to pick it up, the velocity of these red blood cells increase. And so therefore, the 0.75 seconds goes down. And so it's going through, it's equilibrating, and it pops out when they're exercising before there's full equilibration. So now maybe instead of 95% saturated, it's only 85% saturated, let's just say. And so what you get there is hypoxemia because the red blood cells sped up, but this happened primarily because there was some sort of a deposition, interstitial deposition in here that prevented this oxygen from diffusing into the pulmonary capillary. And there you'll see an increased AA gradient. Capital A standing for alveolus, lowercase a standing for artery. So that increased AA gradient you'll see, and we'll show you how to calculate that in a bit. Here's a equation for the AA gradient, and this kind of has some of the items in it that we saw before. This part up here refers to the capital A. That's the amount of oxygen in the alveolus. This here is the smaller case a. This is the amount of oxygen that is dissolved in the blood. This is the PaO2 that you pick up on a blood gas. So let's plug in some numbers for somebody who's normal. FiO2, in this case, would be 0.21. P atmosphere, let's say this is at sea level, that would be 760 millimeters. The P of water is about 47. And the PCO2 would be 40 divided by 0.8. Okay, so let's go through, and let's say PO2. What's a normal PO2? Around 86. So 760 minus 47 is 713. This times 0.21 is going to give us 150. 150 minus 50 is 100. 100 minus 86 is equal to about 14. So the AA gradient is about 14, which is pretty normal. In terms of trying to figure out or guesstimate what a normal AA gradient would be, it should be around 20, 15, 20 or so. Another way of trying to predict it, some people say, is if you use this type of an equation where you take the age of the patient and divide it by 4, and then you just add 4. So a 40-year-old gentleman, 40 divided by 4 is 10, plus 4 is 14. So that's how you calculate an AA gradient. Now, notice, if for some reason we ran into a situation where there was a diffusion problem, what would we see? We would have to see that the FiO2 would go up, because we'd have to give them more oxygen to keep the oxygen the way it was, correct? This would not change, this would not change, and this would either stay the same or go down. We'd have to give a lot of this to keep this the same. You can see that in situations where we can't get oxygen through, we've got to go up on the capital A to get the same or only a small increase in the little a. And so what I'm saying here is that a large AA gradient means there's some barrier or some problem in oxygenating the tissue or getting oxygen into the pulmonary capillary. Okay then, so let's review. If we look at diffusion, a couple key points that you need to know. First of all, it does respond to 100% oxygen. Again, if we look at our alveolus, and we've got our pulmonary artery and our pulmonary vein, if there's a barrier here, just giving more oxygen will help push it through and get the O2 in the pulmonary vein to come up. So it does respond to 100% oxygen. It has an increased AA gradient. Where do we see this? Fibrosis. Any kind of situation where there's a collagen deposition here in the interstitium of the lung, like in pulmonary fibrosis, especially if they're exercising. And why is that? We're going to emphasize that with exercising because the red blood cells are going through here at a faster velocity, and therefore it accentuates this difficulty in diffusion. But you're going to have a lot of thickening for that to happen. Okay, so that takes care of diffusion. We're going to move on to other ones in the next lecture.
Cube, but no dice. Hypoventilation.
Medicosis Perfectionis once again continued our biology playlist. In the last videos we talked about the anatomy of the respiratory system, the mechanics of inhalation and exhalation, lung volumes. Medicosis Perfectionis once again continued our biology playlist. In the last videos we talked about the anatomy of the respiratory system, the mechanics of inhalation and exhalation, lung volumes, capacities, spirometry, pulmonary function test. We even talked about respiratory acidosis, respiratory alkalosis, and the acid-base stuff. Today, we'll talk about what happens to your body when you live on top of a mountain. Now, let's get started. Please watch these videos in order. Let's go. We've talked about this before, so let's review. The fraction of oxygen in the atmosphere is FiO2. When it enters your lungs, oxygen is now called PaO2, A for alveolar. When it jumps on the blood, in the arterial blood, it's called P small AO2, A for arterial. When it jumps on the hemoglobin, it is SaO2, S for saturation. And then the tissue is going to take oxygen and release carbon dioxide. This is metabolism or cellular respiration. Carbon dioxide is going to jump on the hemoglobin. This is called carbaminohemoglobin. Veins have deoxygenated blood and a very teeny tiny amount of oxygen called PVO2. And then you exhale it. Atmospheric pressure. What is it? Imagine that you're standing here. On top of your head there is a column of air. From here until the end of the atmosphere. The weight of this air column on top of your head is called the atmospheric pressure. If I leave here and I go on top of a mountain, I'm closer to the end of the atmosphere, leaving a shorter column of air with lower weight and therefore lower pressure. And that's why when you go up on a mountain, air pressure goes down. Pressure equals force over area. At sea level, there is more air above you, therefore more force, but on top of a mountain, there is less force and therefore lower pressure. At sea level, what's the atmospheric pressure? 760 millimeters of mercury. And since this is the same as the atmospheric pressure, we call this 1 atm. So 1 atm equals 760 torr, or 760 millimeters of mercury. This is at sea level, but if you go up a mountain, this will go down, of course. So, what happened when medicosis was standing here at sea level versus when medicosis went up there? At sea level, there is your 760 millimeters of mercury atmospheric pressure or air pressure. When I went on top of a mountain, of course, the atmospheric pressure went down, say 580, for example. But when I was down here, oxygen constituted 21% of the atmospheric air, and this did not change when I went on top of a mountain. This is very important. If IO2 does not change, whether you're here or here, doesn't matter. What does change is the P big AO2, which is in your alveoli, because this basically equals this crazy equation, but forget all of this. Forget this, okay? Forget this and forget this. Let's focus on this and this. Now let's write it in a very simplistic manner. PaO2 equals pressure of the atmosphere, or the barometric pressure, which is just atmospheric pressure, multiplied by FiO2, which is the percentage of oxygen in the atmosphere. The percentage of oxygen in the atmosphere did not change. However, atmospheric pressure at sea level is higher than that on top of a mountain. And that's why PaO2 is higher here than here. Put differently, if I decide to live on top of a mountain, if IO2 is not going to change, PB, or the atmospheric pressure, will go down, and therefore PaO2 is going to go down. Translation, the pressure of oxygen in my alveoli has dropped. That's not cool, because this can lead to hypoxia. Hypo means low, oxia means oxygen. There is less oxygen in my body. Sea level versus mountain top. What's happening here? Same FiO2 does not change. Higher atmospheric pressure at sea level versus mountain top, and therefore higher PaO2 here than here. I went to the top of the mountain, this is high altitude, which decreased my PaO2 in my alveoli, which decreased my P small AO2 in my arterial blood. This is called hypoxemia. Oh, this is dangerous because we need to give oxygen to tissue. Well, if we are not getting enough oxygen, at least we can increase the number of red blood cells to try to carry and squeeze by every single drop of oxygen available. Try to deliver as much oxygen as we can because we are in trouble here. So, long term, you will have more red blood cells when you live on top of a mountain. We call this polycythemia or erythrocytosis. This is the opposite of anemia. Next, since I have less oxygen now, well, let me try to breathe more. I will increase my respiratory rate. I will hyperventilate, therefore carbon dioxide is going to decrease because I'm washing out my carbon dioxide. When carbon dioxide decreases, this is respiratory alkalosis. Moreover, when I have hypoxia in the lungs, the part of the lung that is hypoxic will just shut down, vasoconstriction, to leave the normal parts of the lungs with as much oxygen as possible. In other words, reallocation of scarce resources. This is what economics is all about. When you vasoconstrict, you will decrease the surface area in the vessel, and of course, when area goes down, pressure goes up. This is called increased hydrostatic pressure, which can lead to pulmonary edema. Basically, you're drowning in your own water. That was deep. When you have a pulmonary edema, you will cough frothy pink sputum. Why frothy? Because the lung has air. Why pink? Because the lung has blood vessels. When you live on top of a mountain for a long period of time, you will have more red blood cells. We call this polycythemia. What type of polycythemia? Is it relative? No, it's not fake. It's actually true. I have more red blood cells in absolute term, not just relative to plasma. Therefore, we call this absolute polycythemia. Is it primary, like a cancer? No, it's not a cancer. It is secondary. It's a reaction. It's a response to the hypoxia. Okay. Is this an appropriate response or inappropriate response? It is appropriate response to hypoxia. So when you go on top of a mountain, erythropoietin will go up. Who secretes this doofus? The kidney. EPO will leave the kidney, will go to the blood, will reach the bone marrow, will knock on the door of the bone marrow. Hey doofus, make some more red blood cells. We're starving for oxygen right here. When you live on top of a mountain for a long period of time, you will make more red blood cells, called polycythemia. Also, you will increase new blood vessel formation. So I'll make more red blood cells and more blood vessels, called neovascularization. New blood vessel formation. Come on people, these videos are beautiful. Please buy me a coffee. And as the saying goes, 1 cup of coffee a day keeps debt collectors at bay. I don't know what the flip I'm saying. Anyways, go to buymeacoffee.com/medicosis. Now, the oxygen dissociation curve. I have a separate video about this topic. Here is the oxygen hemoglobin dissociation curve. You have PaO2 on the x-axis, SaO2 on the y-axis. This is the oxygen that's floating freely in the blood. This is oxygen on top of the hemoglobin. What's happening at the lungs? Well, the lung is full of oxygen if you're talking about normal conditions. Yeah, that's why PaO2 is not low. It is super duper high right here. Okay. What's the hemoglobin saturation? Well, the hemoglobin is super duper saturated with oxygen because we have tons of oxygen in the lung. Therefore, the blood that is close by the lungs will have tons of oxygen saturation. That's easy. So, at the level of the lung, blood is getting oxygenated. Oxygen is jumping on the hemoglobin. We call this phenomena loading. The hemoglobin is getting loaded with oxygen. Conversely, if you're talking about the peripheral tissue, well, as you see here, PaO2 is low, SaO2 is also low, because this is unloading. The oxygen is leaving the hemoglobin and going to the tissue. This black curve is the normal curve. We can shift it to the right, we can shift it to the left. Here is an example of a shift to the right. As we see, we went from black to blue, it's a shift to the right. So, to understand what happens, we'll take the same point on the x-axis and go up until we intersect with the new curve that's shifted to the right, and until we intersect with the old curve. What happened here when it comes to the SaO2? The new curve has a lower SO2 than the old curve. Translation: when you shift the curve to the right, oxygen saturation decreases, which means oxygen is leaving the hemoglobin and jumping on the tissue. Therefore, oxygen is being given to the tissue. Oxygen is released from the hemoglobin and is going to the tissue. And in literature, the right hand symbolizes giving. We are giving oxygen to the tissue. But when you shift the curve to the left, so the black one is the original one, and the red one is the one shifted to the left. Take the same point from the x-axis and go upwards. Intersect with the original one, intersect with the one shifted to the left. As you see, when you shift to the left, you increase the SaO2. Translation: when you shift to the left, there is more oxygen in the hemoglobin, which means more loading, less unloading. Oxygen is going to the hemoglobin and therefore away from the tissue. Thus, with left shift, the tissue is left behind. The shifters. These are the factors that help you shift to the right, and these are the factors that help you shift to the left. The shifters to the right are increase carbon dioxide, increase hydrogen ion, increase temperature, increase 2-3 BPG, increase altitude. The shifters to the left are the opposite. Decrease carbon dioxide, decrease hydrogen ion concentration, decrease temperature, decrease 2-3 BPG, but instead of low altitude, we just say hemoglobin F and methemoglobin. This is the fetal hemoglobin, this is a disease. Unfortunately, many students think that some patients have right shift, other patients have left shift, when in fact, the same person can have right and left shift in different parts of the body, depending on the demand for oxygen. For example, medicosis is here. Look at my shredded physique. My rectus abdominis is on fleek. If I'm exercising my right arm while relaxing my left arm, who do you think needs more oxygen? Well, the right arm needs more oxygen. Therefore, the hemoglobin in the right arm will give the oxygen to the tissue so that the tissue can exercise. But the hemoglobin in the left arm will not give oxygen to the tissue. Oxygen will remain on the hemoglobin. more loading and less unloading. But how did the hemoglobin here know to unload, and how did the hemoglobin here know to load? Well, because these factors happened here, and these factors happened here. Wow! Explain it to me, medicosis. When you're exercising your right arm, this is metabolism, right?
Official
yeah. Therefore, you're utilizing oxygen and releasing carbon dioxide. So this is factor 1. Carbon dioxide is an acid. It will raise the hydrogen ion concentration, causing acidosis. This right arm is exercising like crazy, raising its temperature. And when you are metabolizing, there is too much glycolysis going on, releasing 2,3-BPG, bisphosphoglycerate. Some people write it BPG, some people write it DPG. I don't care. All of this is happening in the right arm, with the exception of high altitude. And therefore, hemoglobin in the right arm is shifting to the right and giving the oxygen to the tissue which needs it. Conversely, hemoglobin in the left arm is preserving the oxygen to himself so that we can wait until we can give it to the right arm, because the right arm needs more. How do the hemoglobin here know? We have low CO2, low hydrogen ion, low temperature, low 2,3-BPG relative to the right arm. And with left shift, the tissue of the left arm is left behind. Economics is the study of the use of scarce resources, oxygen, that has alternative uses, right versus left arm. See, medicine makes so much sense once you understand what the flip you're talking about. High altitude causes hypoxia, hypoxemia, which causes hypoxia, increased EPO and secondary polycythemia. It causes hyperventilation. It can cause some head swelling and headache, pulmonary edema and cerebral edema, which can cause headache. And this is the most important slide in the entire stinking video. On the mountain top, there's hypoxemia, which means low P small a O2. This causes hypoxia, which means just low oxygen in your body. Hypo, low. Oxy, oxygen. Alright, when I have low oxygen in my body, I will release more EPO from the kidney to increase formation and release of red blood cells from the bone marrow, erythropoiesis, causing secondary polycythemia. Polycythemia is the opposite of anemia. Polycythemia has increased red blood cell count, increased hemoglobin, and increased hematocrit. High altitude shifts the oxygen hemoglobin dissociation curve to the right. You're giving oxygen to the tissue. More unloading, oxygen is leaving the hemoglobin. More dissociation, oxygen is dissociated from the hemoglobin. Less binding of hemoglobin to oxygen, less affinity of hemoglobin to oxygen. All of these are synonymous. High altitude, hypoxia, let me hyperventilate in response. This will raise your respiratory rate, drop your carbon dioxide, and when you're losing the acid, you get respiratory alkalosis. I have like 1,000 videos on YouTube, plus more premium courses on my website, such as my antibiotics course, my kidney physiology course, and my cardiac pharmacology course. Go to medicosisperfectionalis.com. Use promo code NEWYEARLEARNING for a 60% discount. Thank you for watching. Please subscribe, hit the bell, and click on the join button. You can support me here. Go to my website to download my courses. Be safe, stay happy, study.
So from the oxygen flux equation, remember
Ooh, very nice accent.
So from the oxygen flux equation, remember oxygen flux equals cardiac output multiplied by oxygen saturations multiplied by hemoglobin concentrations. From this equation we can see that if we increase the hemoglobin concentration, we can also increase the oxygen carrying capacity of the blood. And this will happen over time. Now, we already know that the levels of oxygen are detected in the chemoreceptors in the aortic arch and the carotid body and carotid sinus. But as well as that, do you know where else in the body that oxygen levels are detected on a longer term basis? No, there's another organ in the body that can detect the levels of oxygen going through it. Well, if you haven't guessed it, I'm not surprised. because it's actually the kidney. So the kidneys are able to detect the amount of oxygen going through them. So the kidneys sample the arterial blood going into them, and they estimate the amount of oxygen in that arterial blood. And if the amount of oxygen in the arterial blood going to the kidney drops, the kidneys respond by releasing an endocrine hormone. Called erythropoietin. Sometimes it's just called EPO. So when the level of oxygen in the renal arterial blood drops, the kidneys respond by producing this endocrine hormone called erythropoietin. Now what erythropoietin does, it's an endocrine hormone, so it's going to circulate all around the body, of course. And particularly it circulates to the flat bones, and it circulates to the end of the long bones. So here we have the end of the long bone. And of course in the flat bones and at the end of the long bones, what have you got at the end of them here? Well, at the end of long bones and in flat bones, you've got the red bone marrow. It's the red bone marrow, of course, that's producing the blood cells. Most of the cells produced by the red bone marrow are the red blood cells. So the erythropoietin stimulates the red bone marrow. And it stimulates the red bone marrow to produce more erythrocytes, the red blood cells. Remember, these are biconcave discs. They're darker on the outside. They're lighter on the inside, because if you look at them from the side, they're biconcave discs. This makes them flexible so that it can squeeze through small capillaries. And of course, it's the erythrocytes that carry the hemoglobin. 90, 98, 99%, probably 99% of the blood, of the oxygen that's carried in the blood is carried by the hemoglobin in the red blood cells. So if there's more red blood cells, that means there's more hemoglobin. What's that going to do to the oxygen carrying capacity of the blood? Well, if there's more hemoglobin, the blood can carry more oxygen. So if the blood levels of oxygen are low, erythropoietin is produced. Erythropoietin stimulates the red bone marrow. In response to stimulation by erythropoietin, the red bone marrow produces more oxygen-carrying red blood cells. This is not an instant response. It's not quick like the cardiovascular and respiratory response, which take a second or two. This is going to take a week or two to develop to a significant amount of red blood cells. But when the amount of red blood cells increase, that's going to increase the oxygen carrying capacity of the blood. That's going to increase the amount of oxygen carried in the blood. And of course, one of the places the blood is going to go is to the kidneys. And if there's more red cells carrying more oxygen to the kidneys will recognise that there is an increased amount of oxygen in the blood, and the kidneys will respond by producing less erythropoietin. That means there'll be less stimulation of the red bone marrow and less new red blood cells will be produced. So this is kind of a homeostatic mechanism, really, for regulating the numbers of red blood cells in the circulation.
You finished talking? Hypoxia. What is hypoxia? What is hypoxia? That's good. I'm going to use that.
So what exactly is hypoxia? The brain depends on the blood to provide it with a constant supply of oxygen. Thus, disruptions to any part of the body that plays a role in blood or oxygen supply can lead to hypoxia. The four primary causes of hypoxia are no blood supply to the brain, This occurs when the blood vessels that supply the brain with blood are completely obstructed. This is extremely rare and usually fatal. Low blood supply to the brain. Low blood supply can occur when even a single blood vessel is blocked or partially obstructed, as often happens with a stroke. This form of hypoxia frequently affects a specific region of the brain, interfering with the functions governed by that region. No blood oxygen. When the body can't take in oxygen, or the heart or lungs can't properly provide the blood with oxygen, the brain and all other organs suffer from hypoxia. This is quickly fatal. Low blood oxygen. When the body can't properly oxygenate blood, often due to illness such as emphysema or a crisis such as a heart attack, the brain gets less oxygen than it needs to properly function. So what causes hypoxia? Numerous illnesses and injuries can cause hypoxia. Those include traveling to high altitudes, especially for people in poor health and for those who quickly rise to high altitudes. Carbon monoxide poisoning, strangulation or smothering. For example, the chokeholds that some law enforcement officers can use can cause hypoxia if held too long. Very low blood pressure, which is usually caused by something else such as a hemorrhage. Smoke inhalation, choking, heart attack or stroke, medical conditions such as heart attack or stroke, allergic reactions that lead to anaphylactic shock, severe cases of asthma, allergies, hyperventilation, and in infants, improper sleep positions or unsafe sleep environments. For example, young babies can be smothered in a crib bumper or get inadequate oxygen supply while sleeping on their stomachs. So how long can a person survive oxygen deprivation? Deep sea divers, people who practice certain forms of meditation, and a handful of other exceptions to the rule may be able to go longer periods than an average person without oxygen. For the average person, oxygen deprivation poses an imminent threat. The timeline from initial hypoxia to death or brain damage varies with the degree of oxygen deprivation. Minor oxygen deprivation only causes damage over time, but true hypoxia triggers near instantaneous damage. Brain damage can begin within a minute or two of total oxygen deprivation. At the 5-minute mark, death of the brain cells and severe brain damage that accompanies it becomes inevitable. Most people will die within 10 minutes of total oxygen deprivation. Those in poor health often die much sooner. Some people may suffer other medical catastrophes such as heart attack in response to oxygen deprivation. Symptoms of hypoxia. People experiencing hypoxia may feel like they can't breathe, struggle to catch their breath, or experience a rapid heart rate as the heart beats faster to attempt to supply the brain with blood. Symptoms include something obstructing the face, mouth, or nose. Increased carbon monoxide exposure can be a problem in enclosed areas. So a person in a very small space whose face is covered may suffer from oxygen deprivation even if they can breathe. Changing in mood or personality. The victim seems confused. Loss of consciousness, including fainting or seizures. Blue or white lips, tongue, or face. Tingling in extremities. Pupils that don't respond normally to light. Not breathing or not expelling air when exhaling. Hyperventilation or grasping for air. The inability to speak. A person who is truly choking may not cough. Treatment for hypoxia. Hypoxia demands emergency treatment. Don't try to treat it at home or wait to see what happens. Even suspected hypoxia requires a call to 911 and an emergency room visit. As you wait for help to arrive, a technique called the Dobkin technique may slow brain damage. It can even save a person's life. To use Dobkin, place ice-cold water or a cold compress on the face and eyes of the hypoxia victim. The compress must be below 58 degrees and must remain on the victim's face until help arrives, or unless a medical professional tells you to do otherwise. In the immediate aftermath of hypoxia, medical professionals focus on providing oxygen and addressing the cause of hypoxia. For example, a stroke victim may need surgery or medication to clear blood vessel blockage. Hypoxia prognosis. After oxygen is restored to the brain, the prognosis varies. Longer periods of oxygen deprivation, particularly in babies, young children, and seniors, typically produce more damage. There is no single treatment that can cure or reverse brain damage. Doctors also don't fully understand brain damage, so it's impossible to make reliable predictions. Some people will recover fully, others unfortunately do not. A number of strategies can support recovery. They include addressing the underlying cause of the hypoxia, supportive therapy, including physical or speech therapy to regain brain functioning, preventing future cases of hypoxia, providing psychological support. When hypoxia is the result of someone else's negligence, a lawsuit can aid recovery by providing funds for high-quality medical care. It may also help prevent this from happening to you or a loved one again. For additional information, please continue reading spinalcord.com.
Certain agitation, right? A is going to be
fuck my stomach. Too much coffee. Not enough coffee. Well, it's past the top of the hour.
Is it lower?
I've got to say, this should be all that I have to give you for tonight. Farewell, may we meet again.